Today we're going to be taking a look at mutations. For this video, it's specifically going to focus on gene mutations. Before we get into that, let's review these terms again. So here's a chromosome, and a chromosome is basically just DNA, and it's wrapped up together, and it makes this structure right here. Now you can see there's different parts here. There's like red and blue parts to the chromosome. If we zoomed in and took a look at those different parts, here's the DNA. And you can see that the DNA is divided into different sections. We call those sections genes. And this gene, let's say, it gives information for making a protein, and let's say that protein helps with the color of your hair. This section right here is a different gene that gives information for another type of protein, let's say a protein that helps make your eye color. So those are genes, just different sections of a DNA. Now, if we take a look at this one gene and zoomed in even more, we can see all of the different nucleotides. Now remember, a nucleotide is made up of three parts: a phosphate, a sugar, and a base. So you put this one nucleotide together with many other nucleotides and you get this DNA structure. Now, we talked about how the way that our body reads our DNA is in groups of three. We call those groups of three nucleotides, we call them codons. Today we're going to be taking a look at mutations, and mutations are just any types of changes that can occur in your DNA. I don't want you to think that all mutations are bad. Sometimes mutations or changes in our genetic material can be good, but most of the time it has no effect. Now, how do these mutations or changes occur? Well, they're really just random. But sometimes we expose ourselves to things that are harmful, like chemicals or radiation, that can really change our DNA. But most of the time, mutations or changes in our DNA are just strictly random. We have no control over it. We're going to take a look at mutations in the gene. So specifically, something's changing within these bases. It could be the T's, C's, A's, or G's. Something's changing. So here's our first gene mutation. This is one gene, just a section of the DNA. And then here's the mutated version. Now if you compare the bases, okay, we're going to find something different in the mutated version. So here we have a thymine in the original DNA. There's also a thymine. Two adenines. There's also an adenine here in mutated. A cytosine in the original. A cytosine in the mutated version. Here's where things went differently. Here it used to be a thymine. Now it's a cytosine. And in this situation, we call it a substitution, where one base is changed to a different base. Now, if you take a look, everything before this change is the same as the original. So here we have TAAC. The mutated version also has that. Everything after the change, GCA, GGTT, is still the same in the original DNA. The only thing that changed was this one base. So here's an example, and I want you to do this in your notes. You're going to highlight the first base on the mutated DNA that was changed to show that it's a substitution and mutation. If you see, the A is changed. It used to be a G, now it's an A. But everything before on the mutated DNA is the same exact as the original, and everything after the mutated DNA, after that substitution, is the same as the original DNA. This is called substitution. This is a different type of gene mutation, and it's called insertion. Now if you take a look and we're comparing the original DNA to the mutated or changed DNA, something is different. Here, take a look. A C is added. Now this is not substitution, because if you see, everything afterwards, it just got shifted over. So here we have TGC, and I still see that, but it's just moved over, TGC. Afterwards, we have AGG, and it's still there in the same order, AGG. And then the last two bases, T and T, and then here we still have T and T. So here, this is different. This is called insertion, where a new base is added. Okay, and I know that this is different from the last one because after I added the C, everything afterwards just got shifted over. This is called a frame shift mutation, okay, because everything afterwards just gets moved over. So take a look at the example, and I want you to highlight the first new base on the mutated DNA that was added to show that it's insertion. If you see, the A was added, and I know that the A was added and it's just not a substitution, is because everything after the A just got moved over. So here we have the codon or group of 3 bases GCT, and here we still have it GC, but it moved over. T is on this codon now. The next one is TAC, and here we still have TAC, but it's just shifted over. Here's a clue to help you tell the difference between insertion and substitution. In insertion, if you see a leftover base here, that means somewhere on the mutated DNA a new base was added. So if you see a leftover base, a new base was added. The last type of gene mutation is called deletion, and it's also a type of frame shift mutation. Now here we have the original DNA and the mutated DNA, and the thymines and adenines and cytosines are all the same. The thing is, here there was a thymine, but the thymine got removed. And everything afterwards got shifted over. So by taking out this thymine, the next group of 3 letters that we have, GCA, it's still there, GCA, it just got moved over because the T was taken out. The next one was GGT, still there, GGT, and then the last base, T. So in deletion, we remove one base. Then everything gets shifted over to the left. That's why it's called also a type of frame shift mutation. So take a look at the original DNA and the mutated DNA, and I want you in your notes to highlight the base in the original DNA that got removed. If you see the G in the second codon got removed. And I know that because everything afterwards got shifted over to the left. So here we have AGC, and because we took the G out, everything just shifted over here. We still have AGC. Here we have GTG, and here the codons here together. Here we have CTT, and here there's CTT. And here we have AC. Now here's a clue. The difference between insertion and deletion is if you see on the mutated DNA that one base is missing from the last codon, that means that's a clue for deletion because a base was taken out. So now you're going to do 3 practice problems, and here we have the original DNA and the mutated DNA, so something changed in the mutated version. Now what you're going to do for each DNA, you're going to transcribe it. That means you're going to take this DNA and turn it into RNA, okay? Then you're going to translate that by using the amino acid chart. Once you're done doing that, you're going to tell me which one is this type of gene mutation shown. Deletion, substitution, or insertion. And then you're going to highlight the amino acid. So something on the translated mutated RNA. What is the amino acid that changed? So highlight it and then tell me, was it a frame shift mutation? Did everything shift over or not?